So first of all, before I kick off, I would like to thank Gordon and everybody, the sponsors, for this wonderful meeting. I think it's a nice opportunity to present something about our research about tumor-associated carbohydrate antigens, or also known as TACAs, in colorectal cancer. And before I start off, I would like to thank Katarina Madunic and Di Wang because they did most of the work. And as the title already suggested, I will be talking about colorectal cancer. And just to give you some numbers about cancer worldwide, we have around 20 million new cases in 2020 and around 10 million deaths in 2020. And if we look at colorectal cancer itself, it's the third most common one in GARD in incidence, but then actually it's the second most leading one to mortality. So what is actually going on with colorectal cancer, and why is there an urgent need for new early stage biomarkers and therapeutic agents? Well, if we look at colorectal cancer, what we see is that diagnosis is really normally at an advanced stage. And due to that, there are normally quite some unsuccessful treatments. And also one of the issues is that the tumor is quite heterogeneous. So what we would like to do is to look at the glycosylation. And I don't think I need to tell the audience over here that we know that glycosylation changes in cancer. So that's what we wanted to investigate more in depth. So the aim of the study was to identify if we can find specific tumor-associated carbohydrate antigens for colorectal cancer, and we did this for the N-glycome and the O-GalNAc glycome. And then also what we wanted to know if we can determine if glycosylation features are affected by the tumor microenvironment. So how do we want to do that? That's by using laser capture microdissection. So by using laser capture microdissection, we can really cut out specific regions of interest if we have really heterogeneous tissues. And this is just an example. I don't know if you see it in the back, but we have some green lines over here, the specific regions of interest that we can cut out. And in that case, we can have tissues that are more like normal. We can have the tumor tissue, but we also can look into stroma and see if the glycosylation changes in the different tissues. So the complete workflow that we've been using, is FFPE tissue. Then we did the LCMM. Then what we do is we homogenize the cells and then do cell lysis, antigen retrieval, and after that we immobilize our sample on a PVDF membrane. Then what we can do is PNGSF release, so we release the N-glycans and we elute them from our PVDF membrane. And after that we can also still release our O-glycans using beta elimination and elute those from the sample, from the plate. And then we use PGC LCMS for the analysis. So this is quite a high throughput method because we can do it in a 96-well format, and we need around 20,000 cells, so it's quite a sensitive method as well. And the benefit is that we can not only just study the N-glycome, but we can also study the O-glycome. So why do we use PGC LCMS, so porous graphitized carbon? And that's mainly due to the fact that it has a really powerful separation. So what you can see here from a CACO2 cell line is that if we do an extracted ion chromatogram of a specific mass that should be related to a glycan, we observe two different peaks. And by using PGC LCMS, we measure this in negative ion mode. We get specific cross-ring fragments that are unique for the specific isomers. And due to that, we can really distinguish which glycan is actually belonging to which peak. So it's powerful in isomeric separation, and we get these cross-ring fragments. So going back to our study, so just to show you an example how such an N-glycome profile looks like. So on top we see adenocarcinoma, below we see normal colon. So you can see that this region is rather similar, but if you look over here, we see quite high diversity in normal colon compared to adenocarcinoma. And to dive a little bit deeper into this, actually what we observed is that around 60 N-glycans are shared between cancer, normal mucosa, and stroma. And with that, actually 23 N-glycans were unique for cancer, so they were not detected in the normal mucosa at all. And looking at our glycans, we can of course look into the type of glycans that we have: complex, hybrid, monoantennary, oligomonocytic, and polymonocytic. But we could also look into cellulation. So what you can nicely see is that we have some alpha-3 cellulation popping up for cancer, and not so much for the alpha-6 cellulation. Another way looking at this is, of course, using a principal component analysis. And actually, we were quite surprised by how nice the results are. Because what you can see is that with the normal mucosa, we see a nice clustering of those samples. We see a clustering of the stroma, as well as of the cancer. And if we then look at the loadings plot, what we can see is that the normal mucosa is actually really clustering based upon bisection, so the derived trait where we see bisected glycans. Another feature that we observed is that actually the stroma tissue showed a lot of core fucosylation and fucosylation. And then the monoantennaries were more abundant in cancer. Another way to look at this is really going more in depth. So on top here, I think it's a bit hard to see in the back, so I'll try to walk you through it. So what we see here are the derived traits. So we have acylation, monoantennary glycosylation, and then here we have the unique glycans. And these are upregulated in cancer, and these are downregulated in cancer. So what you can nicely see is that bisected N glycans are indeed downregulated in cancer, where we have monoantennaries being upregulated. So, and then, sorry, we went too quick. Yes. So if we look at previous findings, so there's this nice review from 2015, so it's a little bit outdated, but still we looked, okay, how does our results relate to the previous findings? And what we observed is that indeed we see complementarity between, or we see that it's in agreement with the bisected N glycans being downregulated in cancer. However, upregulation of high manos or core fucosylation is not what we really observed if we compared cancer versus normal mucosa. But then actually, what we noticed to see is that if we compare cancer versus stroma, there we see that the oligomenocytic and monoantennaries are actually upregulated. So that's the benefit of using LCM, where we can really capture specific regions of the tissue, where we can, and with the previous studies where this was all just one bulk of sample. So this was interesting, and then looking more in depth in the high sensitivity for colorectal cancer, 13 glycans were not detected in normal mucosa, but at least appeared more than in 2 of our cancers. And then glycan E, so this one, the diantennary, appeared to be the highest, to have the highest sensitivity with a frequency of 39% and no expression in normal mucosa. But what was actually interesting to see is that if we combine a panel of 5 glycans, then actually we get a really high sensitivity of 89% with no expression in normal mucosa. So of course, it was also mentioned in the previous talk, it's hard to develop a panel that we can target 5 of these glycans. But it's already nice to see that we can find some high specificity for colorectal cancer. Now moving on to the O glycans, actually there our chromatograms are a little bit more clear. Here you can nicely see that the CL2 antigen was upregulated in cancer and not present in stroma, for example. Also here, looking at the PCA plot, we see that normal mucosa is nicely clustering together, stroma is clustering together, but we see a little bit more variation in the cancer. And that is mainly related to the fact that we had 2 mucinous adenocarcinomas in there, so T3 and T2, and those showed a very diverse glycosylation and very heterogeneous glycosylation compared to the other tumors. So it was not that surprising that those were popping out in our PCA plot. Then in regard to how many O glycans we observed, and what was strikingly is that we observed 100 O glycans to be specific for the tumor. And of course, these were mainly due to the T2 and T3. But 100 unique O glycans that didn't appear in the control sample or in the stroma. And then if we dive a little bit deeper into that, what we've noticed is that actually COR2 was the one that is most upregulated in cancer, and not that much, it's downregulated in normal mucosa. And then actually if we combine it with the Shield-Lewis XA motif, then actually we also see that it's upregulated. But that's just in combination with a COR2. If it's combined with a COR3 structure, actually it's downregulated in cancer. So that is quite interesting to see that COR2 seems to be rather specific for the colorectal cancer. And then also if you look at the specificities we had 7 glycans that were really showing a high specificity for cancer. We had glycan A with a 72% frequency and no expression in normal mucosa. Then we had glycan G that also had a 72% frequency but only appeared in one of the normal mucosa samples. But then actually by combining these 2 glycans, we got a frequency of 94%, so really high sensitivity with only one appearance in normal mucosa. And these, what was also interesting to see is that they show an even higher expression in invasive cancer versus non-invasive cancer. And of course, it's always the question, okay, did we make the assignments correct? So what we've also done is that we chemically synthesized the standards to make sure that we indeed have the few codes on this arm and that we had the salicylic acid on this arm, for example. So what we want to do later on is that we have our TACAs as promising targets for cancer immunotherapy. Because why are glycans important? Because they're expressed on the cell surface. And what we assume is that these targets are expressed by many proteins. So normally we have immunotherapy that is really targeting a specific protein. But if we can target our glycans, which are expressed by many proteins, we might have a more efficient therapy purpose. In addition, there's a poor immunogenicity of glycans alone, and there's a limited specificity of antibodies targeting glycans. So this is all really promising, and actually we received funding to dive deeper into this indeed to see if we can develop these antibodies for these specific glycans. But another part that I would like to highlight, because I just started a new position at the University of Groningen, is that we want to also focus on single-cell glycomics. And we actually started this work at Leiden University Medical Center, and where we looked whether we can reduce the number of cells that is needed for the analysis. So now we need 20,000 cells. So it's a small amount, but still there's a chance that you have quite some heterogeneity there. So for this purpose, we have developed a single-cell glycomics workflow where we immobilize. So in this case, just to explore what we can do and how far we can go, we used a cell line. Then we had different amounts of cells. We immobilized first we lysed the cells, we immobilized them on the PVDF membrane. Then we released the end glycans. We add an internal standard to also monitor how sensitive we are. And then we elute our end glycans. And in this case, we actually moved from a different separation platform. So we went to capillary electrophoresis, and we wanted to measure it in normal polarity, so we needed to introduce a cationic label. Yeah? No? Yes. And then we used the CEMS to separate our glycans and to detect them. And to just show you some results is that from a Patu-S cell line, we used 5,000 cells as a starting point, and the most abundant glycans that we observed were the high mannose types. And what we should note is that the main disadvantage of using CE is that we have a loading issue. So we only load a minor fraction of our sample, so which is roughly equivalent to 7 cells. But we still observed a really nice profile. I just show here the most abundant ones. But we could go down from 500 cells to 50 cells to 5 cells as a starting point. And you do see that the profiles get a little bit more messy, but we still observe them with a signal to noise above 9. So after this, we were thinking, can we really push it to this 1 cell? So, and also to show you what we have done and what actually, what you should take into account, of course, is that if you have a lot of cell numbers, you see a lot of glycans. So we saw more than 200 glycans. If you go down, it's nice, but you see less. So you only see the most abundant ones. So there is always to find the balance, what you still want to see. But we improved our method a little bit more. We can now inject 10 times more, which should, in theory, provide us that we can use 1 cell as a starting material. So this is really preliminary data, but I'm happy to show it here today, is that indeed with the 5 cells now, with a 10 times higher injection, we get nicer profiles, but we indeed got also a profile from 1 cell. So it's in theory, it's from a high bulk and we diluted it, but I think it's a nice starting point to go really towards single cell glycomics. We also tried it with a different cell line and we got similar results over there. I must note that Of course, not all of our replicates showed something, because it's always the question, do you really have one cell in your file? And we don't have the tools to really look into that yet. So that are things that we will be working on in the upcoming years. So with that, I would like to conclude. So I hope I could show you that in regard to the TACAs, we really found specific differences for colorectal cancer between stroma and normal mucosa. We had a really high specificity in regard to N-glycans and O-glycans. We had downregulation for the N-glycans for the bisection. And for the O-glycans, we really saw that the core 2 O-glycans were upregulated for cancer. And as a future perspectives, I already briefly mentioned, but we want to develop new antibodies to really target the specific TACAs that we have seen. But we also need to explore if they are expressed in other tissues, other cancers. But also to see whether or not indeed there are specific proteins expressing these glycans, or that it's more general. With that, I would like to acknowledge our group at the Center for Proteomics and Metabolomics, mainly Katarina Madunitz and Di Wang for the glycomic research, and then Wenjun Wang for developing the single-cell glycomics workflow. And just one side note, as I'm starting at the University of Groningen, we have a PhD vacancy opening up, as well as 2 technicians. So if you have someone that might be suitable for a job, please let me know. And thank you.